Before we start, I'll just like introduce myself and some of the co-panelists here. My name is Nathan Salamonis. I'm a professor of pediatrics um, at Cincinnati Children's in the Division of Biomedical Informatics. And I'm one of the MPIs of the LungMAP Data Coordination Center. With me here too is Josh Fortride, who oversees um, all data-related issues within our data coordination center. Josh?
Unmute. Yes, I'm Joshua Fortrade. I work in the LungMAP Data Coordinating Center with the data submissions and data ETL. I work with the investigators to make sure that we get the data and then get it shepherded into our various systems.
And also on with us is Pankaj Rajeo. Pankaj, you want to talk about yourself and your in LungChat?
Yeah, so hi everyone. I'm Pankaj Rajdev. I'm a third year PhD candidate at Cincinnati Children's Hospital in biomedical informatics. Work with LungMap DCC on various kind of data projects. The main projects that we are going to present today is LungChat, which is a multi-agentic framework for talking to your single cell and spatial transcriptomic data without any coding required and generating new hypotheses with it. So yeah, excited to present that today.
So first, I have nothing to disclose, no conflicts of interest or conflicts to report. I first want to start off with an introduction to what LungMAP is and some of the resources that are available. So LungMAP is an ecosystem of different tools, platforms, and interfaces for deposition of data. So we have what's called an ingest broker in which you can contact us and we can give you access to this ingest broker. This allows you to actually contribute your own data sets to LungMAP. LungMAP contributors are primarily LungMAP consortium members, but we accept submissions from the outside. These data sets are provided in various interfaces and endpoints with both raw and processed data available. Some of these endpoints include being able to reanalyze data sets directly in this online cloud platform called Terra.bio. There are robust tools for browsing these data sets, as I'll show you later, the LungMAP data browser, image-based analysis ecosystem called Omero, a bunch of tools that link out to directly from LungMAP, including LungChat, we'll be focused on today, along with a series of other web browsers, standalone web browsers, and community portals like LGA and TopCell, which you may have heard of. The major data types in LungMAP are genomics, imaging, spatial omics, and proteomics. Spatial omics can include spatial transcriptomics, spatial imaging, and even metabolomics and lipidomics data sets. There are a variety of data sources. Primarily, our research centers, or RCs, provide most of these data, along with our human tissue core. But we obtain a lot of data from our community. We're developing LungMAP phase 3 in our current phase, disease portals. And those disease portals are attempting to aggregate large numbers of resources from diverse laboratories across the omics spectrum and metadata spectrum to provide in both separately and in an integrated fashion to the research community. And we're also obtaining data from other sources like dbGaP, GEO, and ERA. We try to adhere to a what's called a fair metadata model in which all of our data sets are aligned where possible to standard ontologies, cell ontologies, sample ontologies, disease ontologies, developmental assay, and so others. We also primarily are focusing on a metadata standard from the Human Cell Atlas Consortia, where we try to align all of our experimental data to the schema. It's a well-established schema. And try to align our cell annotations through an initiative called Cell Cards, which LungMAP is one of the leads of, that standardizes cell annotations for lung cell types, and have been more recently developing AI platforms where we distribute much of this metadata in addition to that through our central website, lungmap.net. And there are a number of tools we're developing. These are standalone tools for, for example, single cell data analysis, pathway analysis, cloud-based analysis of data, but more frequently, generative AI that we'll be talking more about today. If you want to go to LungMap, you would start typing in lungmap.net. That would lead you to the website, which this is a snapshot of. The lungmap.net website has a variety of interfaces here. But if you want to start off with viewing datasets, you can select on View All Datasets. If you want to access standardized, harmonized information about lung cell types, cell type definitions, markers, imaging data, and omics data, please start with lung cell cards. And if you don't know where to go, but you really want to understand more about using these resources, you can start with tutorials. Again, if you want to access these datasets, you can click on these links on the homepage. These are some highlights, recent highlights from our workflow. Or you can start with our semantic search. These can be disease queries, these can be technology queries or cell type queries or specific browsers. Selecting that data option will lead you to a number of different studies. We're showing here all the single cell RNA-seq studies. This is an older snapshot. I think we have far more single cell studies now. Bulk RNA-seq, microRNA, proteomics, metabolomics, lipidomics, methylation, and a large collection of 2D imaging data, including spatial transcriptomics. If you select one of these links, this top link, for example, you'll get a description of a data set. You'll obtain link outs to the original publication, geo-accession numbers if available, or other link outs to other databases, along with a large number of exploratory tools. So these include something called the Shiny Cell Browser, which lets you interactively explore the single cell data, matching cell cards terms for the cell types they're aligned to, something called an azimuth browser I'll get to, JBrowse, and others. If you click on the Shiny Cell Browser as an example, here's this data set for bronchopulmonary dysplasia from Shin Sun's group at UCSD. You'll obtain this UMAP view to start off with, where you can query specific genes collectively across the entire data set. You can also subset to specific cell populations or specific conditions, and only view gene expression or cell types within very specific conditions or samples. For example, here we go to this violin plot view within the Shiny Cell, and you can just select a single cell type, in this case, this SCMF, which is only present primarily in fetal development and at birth in humans. And you can look at differential gene expression within these cells in different babies in this case, whose lungs were taken post-mortem from babies with active evolving BPD, where you don't have expression of this particular gene, ZBTB16, versus controls where you have abundant expression within the cell type. And so this really gives you a nice way to basically view individual cells level data expression across individual cohorts and individual conditions. And this is a really flexible interface for varying the different parameters you want to view with a number of other plots, including proportion plots, heat maps, and bubble plots. If you want to basically see any data set with a Shiny Cell, you can just click on the Shiny Cell link here, or you can type in Shiny Cell or Shiny from the search bar, and it will lead you to any data sets here that have Shiny Cells, for example. And so you can select any of these data sets and go directly to their Shiny Cells or other browsers. You can also select individual genes, for example, or type in any gene of interest, and selecting a gene will let you basically see, is that gene a marker of well-defined cell types? So these are our cell cards definitions. It will also basically link you out to any data sets in which that gene is a marker in. And so these are the mouse data sets and human data sets. So here you actually, if you select this ACTA2A and LGA, or over here for human, it will lead you to this browser where you can view any data set in which this gene is a marker in the LGA platform across mouse or human development or disease with these high-level summaries. Similarly, if you select on the top cell link, you'll be able to access all the data sets in which this gene is a marker in. You'll get immediate information about what cell types it's most predominantly expressed in as a marker and conditions. And you can go off and you can select any of those genes, those links, and view heat maps and gene set enrichment information. You also have access to antibody product numbers that are used in the spatial data that we're generating, along with nucleotide probes. If you select on one of those antibodies, you'll actually get a link out directly to all images. These are high resolution images, confocal, often with many different Z stacks that you can navigate to and change the colors on through different developmental conditions or disease. We also have a robust epigenomics browser. And so if you click on the epigenomics browser, you can view attack-seq data, for example, from healthy human lungs at various time points of collection for a variety of populations and look to see potentially where enhancers or other regulatory DNA sequences reside in the genome that correspond to open chromatin. This is the cell cards browser. The cell card browser currently has approximately 100 different cell populations to navigate from. Each of these cell populations here, we're selecting AT1, has these detailed definitions, descriptions, functional descriptions, definitions, disease associations, along with reference data sets in which the different marker genes for this cell population are described in those reference data sets with link outs or annotation collections, along with images. And if you select one of these images, they'll link out to this resource called Omero, which I mentioned before. Here you can control which colors you're seeing for these different cell types. These are very high resolution images, so you can zoom in at a subcellular level, change these plots and change these colors, for example. Another tool that I mentioned before is Azimuth. This is if you have your own single-cell RNA-seq data, for example, you can just click this browse button and upload it. You can also use this demo data set and align to our cell ref annotations. These are basically cell cards, standardized cell annotations that the lung research community has agreed on that these are these cell populations and these are the defined markers of these cell populations. You can once, after loading your own single cell RNA-seq, in this case, a .h5 file from a 10x Genomics, which is the typical format, you can adjust these quality control parameters. You can select different annotation levels to align to. And once you align your data, you'll get these plots showing the distribution of your cells versus those in the reference atlas. You'll be able to view the expression of individual genes, both at, for example, at a violin plot level or a UMAP level, and be able to explore individual markers within those data sets. You can explore the data at more granular levels. So if you explore these data sets or click on this Omics data browser link in these navigation pages, it'll link to this facile browser, which has this standardized human cell atlas ontologized information. So now you can find all the projects, for example, you can get all the male samples out, you can get all the babies that are of a certain developmental stage or adults of a certain developmental stage or disease stage or a specific technology version. You can filter for those in all these studies, and you can actually export that aggregate collection of data sets, the raw data, directly to Terra instance, for example, or download the files individually if they're not controlled access. If they're controlled access, you need to have dbGaP authorized access to LungMAP, which is a very simple process for requesting access to LungMAP. And you can request those links, and then you can go, basically your data will populate in this cloud repository in Terra, and you can do integrative analyses of that with even your own data sets online in a very cheap space. There are also more recently spatial transcriptomics data sets. So if you search for Xenium here or go to the spatial imaging link, you'll be able to see these, for example, IPF and healthy Xenium data sets from the Banovich lab. This includes, again, data set description along with individual browsers for each one of these individual samples in which this demographic information is shown below. These include interactive heat maps, segmented cell populations, gene expression visualization on the larger spatial plots or UMAPs, drill down to specific cell populations. You can view individual expression of genes, for example, as violin plots or as other plots, and look at the expression distribution on these plots for individual genes. We have a large growing collection of data sets. These are just data sets we've added in this last year. And there's more coming online very soon in terms of other diseases and other disease models. But there's a lot of data that's available. However, today I'd like to primarily focus on this resource that we've been working hard on for the last year or so, which is called LungChat. It's an online platform that acts just like similar to ChatGPT, But instead of asking broad questions about biology, you can directly interface with large, vast collections of omics data. And the goal here is not, pre-processing of data like I showed you with Azimuth, where you basically perform QC, add clusters to your data. These will be possible in the future, but really to take the wealth of knowledge from, in this case, well over a dozen different diseases, spatial and single-cell transcriptomics technologies, and be able to interactively query these and reason across these to develop disease hypotheses. So we're trying to ask higher level systems biology questions to obtain insights here. There are a number of functional capabilities within LungChat. This includes some comprehensive single cell analyses, spatial transcriptomics and integration. So we're not just viewing spatial transcriptomics data here from Xenium, spatial imaging data from Xenium and spatial transcriptomics from Visium, but we actually have direct integration between these. And so we basically align spots between Xenium and Visium. So you can ask, in which spots are these genes co-expressed across technologies and effectively have multimodal spatial queries across these two different technologies on the exact same sample. And there's some more robust integration and stability analyses we've done here. You can perform interactive functional enrichment analyses and perform literature synthesis. There's a variety of backend tools here that are quite novel and impressive. These functional enrichment not only include gene ontology, but include drug targets, transcription factor targets, and a variety of others, including to canonical pathway and MSigDB enrichments, for example, which you can specify through chat. This LungChat platform can directly interface with lungmap.net and ask questions about specific donors, what sample types and data types exist for different donors, which data sets and technologies exist, or where cell types are present across. So there's this robust API connection to LungMap where there are questions for data visualization. Basically, in addition to hosting these large single cell data sets, that we have well-established Python and R code for dozens and dozens of different functions. These basically enforce reliability in the outputs, so we're not subject to hallucination. You ask a question, that question basically generates something called a JSON file. That JSON file then is a record of exactly what code was run, how it was run, and exactly what was run. This enables you to reproducibly reproduce queries that you've generated in the past by downloading these files and evaluating exactly what parameters were executed in this code, and even going back to the original code if you want to and execute that original code. However, in some cases where there is a need for custom code, and so custom code can be generated on the fly through these models to generate custom visualizations. We generally don't focus on those because we want to focus on the reproducible code, which is not subject to AI-based hallucination. And so reproducibility is a central component of this ecosystem. There's PDFs, PNGs, text files, JSON, logs, and interpretation of that data that's generated at pretty much every step in an analysis. This is a multi-agent based architecture. There's a single cell agent, a spatial agent, a supervisor agent, pathway analysis agent, LungMap API, and more advanced reasoning. We execute multi-point orchestration to generate precise literature, basically accessing PubMed at a much deeper level than existing APIs and ontology-based platforms use. This is an exhaustive search that really lets you go much deeper into the literature than previously capable with these AI-based platforms. The central output of this interface are high-quality publication images that you can directly embed in publications and cite with the code, if you like, the JSON code, or even the raw source code for how these plots were generated. There's a variety of different single-cell atlases here. So we support data right now from the humans HLCA, the Human Lung Cell Atlas, which has an aggregated set of standardized, analyzed 35 data sets from dozens of different research labs, spanning over 2 million cells. We support this HCA fetal data set, 2 different bronchopulmonary dysplasia data sets, and induced pulmonary fibrosis and normal spatial Xenium and match Visium data sets. Again, reproducibility is a central focus of what we're doing here. So I'm actually going to go, before I I'm going to step aside from here for a second, and I'm going to open up I'm going to open up here I think you can, actually, I'm going to just close this and open up this link here, over here. And I think you should see a white screen, hopefully, that says LungChat. And I'm going to show you. So actually, before I get here, I'm going to open up a web browser and just show you what I'm doing and where I'm going to. And so here, we're going to go to chat.lungmap.net. So this is the entry page for a LungMap that you'll see. You'll see some example kinds of just queries and outputs here, along with some more background information on LungChat. If you say try LungChat, what you're going to be asked to do, and I'll just close this for now. But let's say, whoops, let's say I'm going to close this one, so it doesn't want to click on it. But I'm just going to say if you were at UCLA, you can say UCLA, and you'll basically be able to select your university. And basically, once you get there, you'll be asked to authenticate. This is two-factor authentication that you would be normally asked to use at your university. This is using a centralized framework called Duos, which is standardized across institutions. Send a push notification to your phone, for example. Authenticate, and once you authenticate, you'll be presented with your own session here. This is a session that is remembered, so you can go back here and you can access what you've previously analyzed in the past, and perform these different queries. If you want to just start getting going without knowing which questions to ask, you can always just select single cell agent, for example. I've used a number of queries today, so it says I have two queries left after doing a number of queries. So there is a daily limit on the number of queries you can do. It's about 15. But if you really need to do more, contact us and have a discussion. And this will interactively perform a series of analyses. In this case, it's generating dynamically a UMAP plot from a specific data set, which we have pre-computed annotations and differentially expressed genes on. And it's generating a UMAP plot across conditions here. And also let that go. And when it's done, it will basically, the AI will also interpret that UMAP further, and tell you what's going on there, at least what it thinks is going on. So these queries are traditionally fairly fast. They're slightly slower right now. Number one, because we probably have a lot of people accessing the system right now. And number two is that we've actually deployed more agents to be able to answer more and more questions and develop deeper insights into these various analyses. But usually a query shouldn't take more than a minute in most cases, and really a few minutes on the outside. As it's generating its results, by the way, there's this dialog up here on the right. You can see it's actually generating results interactively here. So if you click here, you can actually see that it already has generated the UMAP with the different conditions here. You can download this UMAP here. And it's already generated also other results here. So this is just a second UMAP it's generated. And you have its configuration information. So you can download this JSON file. So this JSON file now gives you the specific configurations and parameters that were run to obtain this plot here, along with the unique ID of how it was generated. And you can generate reports for it. It's still thinking, it's still trying to interpret, so it's finally generated its results. And now it's not only just given you the UMAP, it's actually done a deeper synthesis of this data set. So it's telling you what the proportion of different cell populations are, the different conditions, it's describing them in more detail, and it's giving its overall evaluation of this data set and what it thinks it's seeing. And then they can ask you more questions to begin. I'm going to just show kind of a pre-recorded video to give you a little bit more of a stable response than relying on those two queries that I have left. And so here, this is a typical query that you might ask, is you want to start with, well, which lung diseases are there actually differentially expressed genes computed for macrophages? And in this case here, it may be that there are very few macrophages in some data sets, or little to no macrophages, so you're not going to get disease differentially expressed macrophage-associated genes. But in this case, it's telling you where there are cell states and lung disease disease conditions in which any macrophage-associated populations, saying these can be interstitial, interferon activated or SPP1 macrophages would have differentially expressed genes. You can also ask how many differentially expressed genes are present in those data sets and how many samples those are present between. So basically there's a knowledge base that sits behind these queries that is intelligent enough to know. It's telling you at a high level how many donors, for example, here um, are differentially expressed or to have genes associated with them. In this other case, I've asked for where are cell frequency differences between normal pulmonary fibrosis and COPD patients. And so it's showing me a cell frequency plot. In addition to that, it's actually interpreting the plot and saying where there are more or less cell populations. Again, here we have that code, the JSON code that's generated, along with statistics on the cell frequency and differences, the p-value associations, looking at differences between IPF for COPD versus controls. Here we're asking now to see if we can get differences in differentially expressed genes between COPD, ILD, IPF, COVID, pneumonia, and sarcoidosis in macrophages. So what the program's doing is retrieving all those differentially expressed genes and associated statistics. It's looking for the overlap of those genes among these different disease comparisons, and it's generating something called an upset plot. It tells you the total number of unique genes. It's telling you where there are the most genes that are differentially expressed. And it's showing you the specific overlap of genes between the different disease models here. Again, you can download that TSV of gene associations, get that PDF of that plot, and get the JSON code that was used to derive that, and even paste that back into the interface and ask deeper questions about that data, looking at specific groups of shared genes and doing additional analyses, for example, pathway enrichment analyses or network analyses or literature queries on specific overlapping sets of genes between different conditions or unique genes that are only found in, for example, IPF. In this case here, we're asking to show gene network of pulmonary fibrosis differentially expressed genes in one cell population, adventitial fibroblasts. It's going to produce, the goal is to produce a network, and the network here is using well-defined protein-protein interactions and protein-DNA interactions from a variety of different databases where these different interactions are well established and curated and known, and it's restricting them to differentially expressed genes. It's generating a to-do list here. So the to-do list here is to generate first that network, and after it generates that network, it's going to extract those upregulated genes that I've asked it to look for and to identify among those upregulated genes which genes are most enriched for drug targets. And specifically, I'm saying from top gene. So top gene is that database which has, I believe, over a different 1,000 different databases of annotations from which it's deriving basically enrichment results from. These include drug target databases that it can perform enrichment analyses from. And then finally, we asked it to find whatever drugs it finds, what is the evidence for those drugs playing a role in an antifibrotic response? So are some of these molecules actually validated to reduce fibrosis in a pulmonary fibrosis disease condition? And so it's telling you the different steps. It's walking through those different steps. And you'll see here up on the upper right here, it's generating a number of results. You can click on this button anytime and see exactly what files are being generated. Look through those results as they're being generated interactively. Look at the configuration files. If there's custom code that's being generated, you can look at that custom code and evaluate that. And you'll see right now, it's actually performing deeper literature searches on these different genes that it's identifying. So it's performing, so one of its hits is curcumin and what is its antifibrotic properties, and specifically looking at the mechanism and its role in IPF, and the same for other compounds that it's found are also enriched among genes that are upregulated in this gene regulatory network. So it's performing more exhaustive research. And you can see that that list of results, the top of the interface is growing each moment. And so some of these queries like this, if you ask these multi-step questions, can take a little bit longer. But the output you'll get is this basically synthesis of results. So not only has it told you, given you a network, and I'm going to actually go back. So this is a really fast output here, and I'm just going to step through it, and I'm going to pause it at different steps here. So the first thing it gave you is it gave you this image, this network-based image where red genes are upregulated, blue are downregulated. We'll see it more in a second here. But it's also telling you more about this network. It's saying there are 328 genes in this network, 886 regulatory interactions that were specific to IPF, adventitial fibroblast, up or down regulated genes. The most of those genes were upregulated, 266. It tells you what the range of fold changes are. These are pre-filtered for certain statistics. And it tells you what the key nodes in this network are. And so it's saying MYC is a key regulator. It has 121 different interactions. The genes specifically that are targets of MYC here, it's interpreting them and it sees that they're associated with cell proliferation and metabolism. HIF1 genes and 99 of them. And interestingly, obviously, they're hypoxia response genes and angiogenesis. This is showing you that the network is not just garbage. There's biological information here. STAT3 with inflammation and fibrosis, Gene D, AP1 transcription factor associated targets, and VEGF-A. You also get a little bit more information here now on the specific drug targets. And so here, there are a number of, from the 277 upregulated genes, it performs this top gene enrichment analysis, and it's telling you about the top 10 enriched drug targets or drugs that are being predicted to be basically could basically be used to potentially therapeutically pharmaceutically regulate these particular proteins if they're upregulated. So curcumin was the top hit with a p-value of times 10 to the negative 88th, along with these other ones, including MEK inhibitors and the specific genes that they're associated with. And then it actually does this literature synthesis now of saying, what are the pathways that these drugs seem to be targeting? What is the literature evidence for this targeting? And comparing and contrasting these results and saying, really, if you were to focus on particular drugs or signaling pathways, which of those you would be best focusing on. Once you get the associated outputs, you can navigate these in a much deeper way. For example, you can go in depth into this network plot and specifically view these interactions and download that plot. You can select the list of actual protein-protein and protein-DNA interactions, and will indicate in that table and download that what interactions are present. You can sort based on p-values and other statistics in that plot. You can download the drug target associations here. So these are the top enriched drugs and their genes. There's TSV files associated with those you can download. And you can download the synthesis, this report of results. This includes those results at the top. The report here is indicating what the relevant studies are and evidence with link outs to high confidence publications. So not any hallucination-based publications, but there's a rigorous identification and analysis of these PubMed articles with specific confirmed contacts for these drugs and target gene interactions and disease contexts, for example. So there's a lot of information that's being extracted in these types of queries. You can also perform just very simple, straightforward data visualizations like show me a volcano plot of gene IP of pulmonary fibrosis differentially expressed genes in one cell population called macrophages. In this case, the interface is going to tell you not only it's going to give you that plot, but it's going to be able to interpret that plot and say these are the top most significantly regulated differentially expressed genes, which it's showing here. It's describing what their functions are, both upregulation and downregulation, telling you a little bit more about the pathways that are associated with these, and saying that what the potential implications of that, along with being able to download those results. Finally, you can perform these more advanced spatial queries. So the spatial transcriptomics and spatial imaging data we have are for two different platforms. One is this Visium HD platform, which was analyzed at a very high resolution, I think 8 microns. And so you can evaluate which genes are most expressed in different cell populations. In this case, this IPF specific aberrant basaloid population, KRT5 negative, KRT17 positive cells. So it's again generating this volcano plot. It's identifying the top differentially expressed genes. It's providing their fold change and p-value. It's associating these with different pathways. And it's telling you more about which of these markers are actually associated with profibrotic activities. You can download these results and navigate them. In this case, we're actually saying, do this also for Xenium. And so presumably, again, since these are spatially matched, the same samples, it should be able to identify whether there are shared differentially expressed genes or unique differentially expressed genes based on these two different platforms, and where do they overlap? Which is the best marker here between these two spatial platforms? And here it will report back that ITGB6 is one of the most upregulated genes in Xenium. Both of these platforms, Xenium and Visium, have ITG6 as a top marker gene, and it's discussing its potential use as a therapeutic target, and specific drugs or pathways that can be used to block it. Um, there are different pros and cons between these different technologies. Xenium is a smaller gene panel. It says 255. It's actually more closer to 300. That was part of our backend prompts. I think it underestimated along with Visium. It's really, you have about 15,000 genes that are expressed. So again, a little underestimated here. But that's actually due to our, it's not the LLM that's making that mistake, but our previous annotations that went into it. It's comparing and contrasting those different target genes for this aberrant basaloid population between the platforms, and letting you, again, download these results. But more importantly, rather than just doing these basic marker queries, you can perform more advanced queries. And so these can include looking to see, well, in this case, we can just see much more simply, this is not an advanced analysis, but just show me ITGB6 expression as a spatial plot. And so this is just going to show you on that both Xenium and Visium matched coordinate plot the expression of this gene, and you'd presume that you see a similar expression distribution in the same spots, and indeed you do. So you now have a plot where you can directly compare expression of any single gene. You can also ask the AI, see how correlated are these two genes between these technological platforms, and it will give you an answer. It performs quantitative analyses and will give you a direct correlation between all spots in this data set, the incidence of them, and which aligning spots are most concordant, or any spots for any genes, which are the most concordant genes across technological platforms. You can ask, and so here's the more advanced question is, which receptor ligand interactions are most enriched in these aberrant basaloid cells? It will look for cell-cell receptor, so look for cell-cell interactions that are enriched. So we'll say, in this case, for these aberrant basaloid cells, which cells are they sitting most adjacent to spatially? Here, one of the answers is macrophages. There are other cell populations as well, but macrophages is one of the most enriched cell populations these are sitting next to. And then it will identify receptor cell ligand interactions that are also enriched. And so there are plots you can generate that show the concordant enrichment of those cell-cell proximity-based interactions using a workflow called SquidPy in the background, and cell receptor ligand interactions using another workflow called CellChat. And so the AI will basically be able to understand your query and generate various plots and interpret these results. And in this case, it's highlighting SPP1. And again, I think it's, I believe it's ITG6 as the B as the interacting partner, the receptor ligand pair. And it's between macrophages and this aberrant basaloid population. And so it's listing some of those results here, and it's generating some of those interpretations, but there's different ways to access that information. And again, you can generate these, as I showed you before, these simple side-by-side UMAP plots, again, which will interpret, especially across disease contexts, to tell you what some important differences between these are. Once you're done with these all analyses, you have the opportunity to synthesize these results. And so again, there's that option up in the upper right-hand corner of LungChat we'll get back to right here. You can generate this report. You have now something like hundreds of actually outputs here that you generated here from this analysis. So once generating this output, it then is going to generate a PDF file. You can download that PDF file. And from that PDF file, it'll give you basically a descriptive synthesis of all the queries we've done today. And it'll say biologically, what are some of the major take homes, given the overlap of these genes, their cell frequencies, their differential expression, their participation in certain gene receptor ligand, different transcription factor target interactions, their overall regulation, specificity in certain cell populations across platforms, drug targets, and receptor ligand interactions. And so this really gives you kind of a way, in a way, to a synthesis and hypotheses or structure potentially for organizing a manuscript, which, along with literature evidence and synthesis of what results and prior knowledge are integrated. And there will likely always be some useful novel information that's gathered here. You can download any of these result files here or download everything. So that if you click that download option here, it will download this big zip file. And that zip file can be extracted, and you'll have all images and code we just generated without having to go through and manually download each one of those outputs separately. So I think that gives you kind of an overall description of the type of resource that LungChat provides and what lungmap.net provides. And I know that there were some hands raised already. And so I'm happy to kind of step through different questions, or myself or Pankaj and Josh, who developed these technologies, to really help you really understand what's going on here. So we have one question in the chat is, can you please comment on privacy considerations for different tools? For example, if how prompts are stored or used to train the models, similarly with uploading and analyzing data with Azimuth. So those are great questions. For Azimuth, I don't recall the security requirements. So that's a platform that we did not develop. That's developed by Raul Satij's group, and we're supporting a Docker container with those data sets. I have never even thought to look to see if we could see which of those H5s, how they're stored and where they're stored and how transient they're stored. That's a great question. I will look into that and see what the security around that is. At LungMap, we have strong language data use agreements within our consortium that constrain how we can use external data sets. But for the purpose of LungChat right now, in the pre-early phases, we have been using questions to refine the model where possible and improve it. But security is an important question, and I'll let Pankaj answer that more, and if there are potential concerns there. Especially the chats are going right now to, for example, ChatGPT and OpenAI. So some of those OpenAI queries are probably accessible by OpenAI. I'm not totally sure.
Yeah. So regarding privacy, actually, all of these analyses are running inside a sandbox Docker container. So your analyses are not exposed to the web. But if we're talking about the LLMs we are using, so we are using Claude model, Anthropic models, and the OpenAI models. And Anthropic and OpenAI already have access to your queries through the API. What we are doing here is, we know that these biological questions are not like there is no personally identified information in these questions. What we are doing is we are also trying to open source the entire code base, so you can run these analyses in-house by deploying the code on your servers, so it never leaves your infrastructure. A part of our one of the future plans of our LungChat framework is to take the questions, figure out what are the patterns, what are the question patterns where the system is failing, and take that data and further fine tune the model so it can answer your questions better.
Great. And we're happy to open this up for additional questions or commentaries you want to say. Please just enter anything in the chat or raise your hand and ask a question. We're happy to unmute you.
There's a question in the chat.
Okay, great. Yeah. So how do you gauge whether the model is failing or not? That is a great question. And so we've been performing benchmarking. And so for a standard set of queries that we have about like 20 or so that are standard queries. And so it's similar to that transcriptional regulatory network inference question we have. If you say, well, here are, what are the gene regulatory networks of adventitial fibroblasts? These should be the hub transcription factors that are predicted to be the regulatory nodes in the network. That's one objective. a biological objective, and these are the specific drugs that are identified for those enriched genes. And so if we run that query 1,000 times, we would hope to see over 90% of the time that we see reproducibility in those response for the individual drugs that are reported, the individual transcription factors that are nominated from that analysis, and even the literature insights. So which of those drugs have prior evidence of being fibrotic agents? And so we could create scores for those different observations and then see in aggregate over thousands of simulated queries again and again in the back end, whether or not those are successful or failing. So we're really doing that for standard workflows right now. And that's the best we can do with prior human knowledge-based annotations. In some cases, we generate results with a workflow and we say, well, that's an interesting result. And we go and we go back to the literature and prior papers and validate them. So a lot of the KRT5 negative, KRT17 positive results that we showed you in terms of receptor ligand interactions and cell-to-cell interactions, those actually have been that specific result has been documented in the literature, and so we're able to get a little bit more information on it. There are lots of other plots we're actually not showing here, so there's a lot of plots. The model is able to generate autonomous code to address questions. We're trying to minimize its use where possible because we don't trust it. It is subject to hallucinations, so it's in a really important point. There was another question, how accurate is the AI generated results and information? Are they generated only from data within LungMAP? And if this AI generated figure, will it violate the rules if installed in an NIH publication? So no. Any results you generate, it's not necessarily, it would technically not be an AI generated result in that the sense that your human query is being translated into a JSON file, and that JSON query is then being used to run code. And so you can cite the code base and the function that was used to generate that code. And then by doing that, you're not saying that it was an AI generated figure. Even if it was, you now have a JSON file. So you have a parameter file. And so it's the same as you running that parameter file through a script. There's no hallucination between that JSON and the code. Both the code is hard coded. And so the plots I showed you, the gene regulatory networks, the violin plots or UMAP plots or everything, those are all documented and they have very specific output names that constrain which IDs correspond to the JSON code. So you have a direct correspondence between the code that was used to generate it and the outputs. So those should not be considered subject to any kind of hallucination. I would not qualify them as generative AI at that point. It's a great question. And something we need to actually bring up later on when we're discussing this approach. So there are updates that are happening on this website right now. So we're constantly trying to improve these models and trying to ensure that they're both consuming enough and not too much in terms of their large language model capacities. So there's always optimization. So we're trying to balance speed with outputs right now. I can log in as a different user, not as myself effectively, so I can come in through, let's go back. So I'm still logged in. But you should be able to log in through, for example, even Gmail. So if you type in Google in authentication, you should be able to log in. But chat shouldn't be disabled in the.
So actually, the thing is, instead, what you can do is you can visit the website chat.lungmap.net, and at the end of the website, if you click on the, let me share my screen. So if you go to chat.lungmap.net, you can see the contact us button here, or you can also see at the bottom. If you click here, it will send an email to the to me and Dr. Nathan. So I'm just waiting for it to pop up. There's some issue. Yeah, here. So just go to chat.lungmap.net, and if you have any questions or if you want to integrate your own data, you can just click on contact us and reach out to us.